Howdy, my name is Ty Botkin, and I am in Dr. Shepard's Ento 320-500 class. And today, we are going to be discussing how using ethanol on Apis mellifera to study alcoholism. First off, why use Apis mellifera? Honeybees, also known as Apis mellifera, are one of the most highly researched organisms due to honey being used in a variety of products. Apis mellifera is a highly socially complex creature and replicates the social behaviors of humans, such as sharing food and labor. This is why they make perfect organisms to study social experiments with. Why use honeybees for studying alcoholism? The honeybee is a social insect that exhibits high complex social behavior and can learn through scent-laden tasks. Also, instead of injecting other species with ethanol, the honeybee will freely consume ethanol on their own accord, making them willing test subjects. Also, since honeybees are more giant insects, it is easier to study their behavioral changes. Why combat alcoholism? Alcoholism in the United States. More than 7% of the United States citizens are affected by alcoholism. That is around 18 million Americans affected by alcoholism. 1 in 4 children 18 years of age or younger is exposed to family alcoholism in some form. Alcoholism-related hospital visits in the United States are 20 to 40%. $185 billion annually will be spent on healthcare due to alcoholism. Alcohol-related disorders are ranked second in the world. The bees used in this experiment. Scientists experimented with honeybees after they consumed ethanol to study their behaviors. According to Dr. Mays, each one of these honeybees were selected was maintained at 25 to 30 degrees Celsius on a 12-hour, 12-night light and dark cycle in an indoor facility. Individuals were collected in small glass vials and cooled at 20 degrees Celsius for 1 to 5 minutes. Then a subject became inactive. It was removed from its vial and placed into a plastic restraining harness where the bees were fed 18 units of 1.8 milliliters of sucrose solution and held for 24 hours at a room temperature before treatment with ethanol. Now, ethanol effects on bee behavior in time doses. Ethanol affects honeybees' behavior in a time and dose-dependent manner. The average percent time honeybees in each treatment group spent engaged in each behavior for a 5-minute interval. According to the graph to our right, we can see that the walking behavior, after ethanol inebriation generally decreased over the interval and showed a dose-dependent response. But only 50 to 75% of the ethanol treatments were significantly different from the control group. Duration of the time when the subject did not move was dependent on ethanol treatment and increased over the 40-minute period. Grooming behavior was greatest for the 10 and 25% ethanol treatments when compared to the control group that received higher doses. Frequency of behavior shown. Duration of the frequency of bouts of behavior during the first 40 minutes after ethanol consumption. We can deduce that over a 5-minute interval, mean frequency and bouts of walking laying upside down, stopped, or grooming increased. For subjects given each ethanol treatment for each behavior, balance durations were compared with Dunnett's test, and frequencies were compared with at least squares contrast. Recovery time from ethanol inebriation and what occurred afterwards were treated in a 48-hour post-ingestion time. During this time, between the 12 and the 24-hour after ethanol ingestion, in contrast with the subjects ingested with the 25 or 50% ethanol required, the 24 to 48-hour recovery indicates significant ethanol recovery time. Ethanol in honeybees and the time it took for the ethanol to decrease over time. Ethanol levels were significantly lower for 24 and 48 hours time points, while compared to the 0.5 hours time point for all treatments. Sadly, this is the saddest part of the research, mortality of the research. Though the bees can handle ethanol, they can die, sadly, due to this experiment. Mortality of the honeybees consuming ethanol increased if they were exposed to higher levels of ethanol. Conclusion of the research: due to Dr. Mays' and team's research, they discovered that ethanol affects significant motor function in invertebrates, how locomotor behavioral skills are affected by ethanol, and provided a basis for future work investigating ethanol's effects on social behavior, learning, and memory.